Hi, I'm Nancy Ford, and I'm a professor in the physics department here at Simon Fraser University. My research focuses on understanding the mechanical properties of the molecular building blocks of our bodies. So you might be wondering how and why a physicist is looking at these molecular building blocks, which are proteins, and why you should care about this. Those are good things to ask. Collagen is a protein, and it's actually the key building block of our bodies. You could think of collagen like Lego blocks that stick together and build up stronger, larger structures, like our tissues, like bones or tendons or cartilage. However, collagen proteins are far more squishy than Lego blocks. And collagen is more like a thread than a block. It's a long, thin molecule that winds up with other collagen threads to make a thicker thread, which can wind up with other threads to make a rope, etc. As we age, we know that these tissues change their mechanics. Our skin gets wrinkly, our bones break more easily. If the collagen is not made correctly in the first place, other diseases can result, like brittle bone disease or fragile skin syndrome. And we want to try to understand how much that relates to the properties of these individual thread-like building blocks. So, we design experiments to stretch single collagen proteins, these threads, and measure how they respond to force. Where does physics come in? Well, we use our training in physics to build the instruments for these experiments. First, we need some collagen for our experiments. We can buy this from a company, which has extracted it from cow tissue or rat tails, or we can prepare it ourselves. Ala grows cells in the lab that produce collagen. They pump lots of it out, and we can get the collagen from there. So she's checking them, she's fed them, she's washing them, looking at them under a microscope, and seeing, yes, okay, they are very healthy, they are presumably pumping out collagen, and so we can use that collagen if we want to pull on. Once we have the collagen, we have to be able to pull on its ends in order to measure its response to force. Now, collagen is far too small to see with the naked eye or even with a light microscope. So what we do is attach it to a much larger bead that we can manipulate and see in a microscope. And in the experiments that we're going to show you today, what we do is use magnetic forces to pull on the bead and stretch collagen. Microscopes look at really small things. So what I've done is made a larger scale model to give you an idea of what's going on. In our experiments, we have beads. That's shown here by this ping pong ball. Inside the ping pong ball, we put a magnet. So I can take another magnet and bring it nearby. And you probably know when we bring two magnets close together, they can attract or repel. So let's see what happens when I bring this magnet close to my ping pong ball that has a magnet inside of it. Woo, and you can see that it's pulling up on the thread on the collagen. So magnets force at a distance, pulling up on this bead and stretching this thread, which would be our collagen in the experiments you're about to see. These experiments are performed inside small microfluidic glass chambers that we can put on our magnetic tweezers microscope. To get started, we have a glass slide that's chemically treated to bind one end of collagen, and the beads have to be chemically treated to bind the other end of collagen. So Janine is preparing the glass in this for these experiments. She's cleaning it, and then she's coating it using different approaches from chemistry so that it can bind the collagen. After she's prepared these, she needs to insert the beads and the collagen so that they can bind to glass. So here she is loading the chamber with a micropipette. Finally, we're ready to tug on the collagen proteins. So Luis and Janine have put their sample into our new microscope, and right now they're adjusting the optics to bring the beads into good focus. You can see that each bead appears as a circle with many rings around it. Finally, we apply force to the beads. So these beads are magnetic, like in my demonstration, so they're pulled upwards when we move a magnet close to them. And the closer they bring the magnet, the stronger the force that's pulling upwards on that bead. We can also rotate the magnet, which causes the beads to rotate. You can't see that a single bead is rotating because it's circular. But if we find two beads stuck together, then when we rotate the magnet, you can clearly see that we can spin these two beads around as Luis adjusts the magnet's angle. All right, so now we're ready to do our experiments on collagen. We've taken you through all the steps. We can raise and lower the magnets, seeing how much collagen extends when different forces are applied. Is it very extensible, like a rubber band, or is it more rigid, like a thread? And once we establish this, we can learn about how changes in the composition of collagen related to diseases or aging alter this response. We want to understand the key building blocks of life in order to help combat disease and perhaps even build new materials out of them that are environmentally friendly and biocompatible.